DNA, or deoxyribonucleic acid, contains all of the hereditary or genetic material in humans and most other organisms. So the DNA is what lets people know what genetic material is in a person or is in a certain creature or other organism. Now each cell in an organism has the same DNA. So if someone were to get a piece of your hair and a fingernail and a piece of skin and look at it very closely and look at the DNA in all three would be the same, and it would let whoever was looking at this know that all three of those articles came from the same person. This is the same for other organisms. Each cell in the organism's body is going to have the same DNA pattern so that you'll be able to tell that it came from the same creature. Most DNA is found in the nucleus of a cell. There is some DNA in the mitochondria, and then it is known as mitochondrial DNA, or it would look like this if you saw an abbreviation for it. So if you see the little MT before DNA in the abbreviation, that's because it's mitochondrial DNA and it's found in the mitochondria instead of the nucleus. But most DNA is going to be in the nucleus. Now, that important hereditary information found in DNA is stored as a code made up of 4 chemical bases. We've got adenine, guanine, cytosine, and thymine. And each one of those bases is only going to pair with one other base. They can't all pair up with any of the other 3. So adenine pairs with thymine, and guanine pairs with cytosine to form base pairs. And a way that I remember that is that the ones with the straight lines in their abbreviation letter, the A and T that can be made with straight lines, go together, and the ones that require a curved line go together. And that helps me remember that adenine pairs with thymine, these straight lines to form A and straight lines to form T, and guanine goes with cytosine because of the curve shape that it takes to make each of those abbreviation letters. The sequence of the adenine, thymine, guanine, cytosine pairs determines how the organism builds and maintains itself. So it's important to think about this. Not every organism or every person is going to have the exact same sequence of base pairs. And you might think of the alphabet. We use 26 letters to form all of our words and sentences, but the way that they're arranged, the sequence of those letters, is what lets us know what word is being communicated, what sentence is trying to be communicated. And each word has a different meaning, even though it's made up of the same 26 letters or some subset of those letters. So it works the same way. You're going to have some sequence of adenine, thymine, guanine, cytosine, and those base pair sequence is going to let you know, or let a scientist know, what that organism does to maintain itself, what kind it is. Is it functioning properly? Is everything in the right order? What does that order tell us? The same way that seeing CAT tells us that that is the word cat, and it's talking about a mammal with pointy ears and whiskers and a tail that's kept as a pet. Let's look at the next part. Each base pair, so our adenine thymine pair or guanine cytosine pair, is going to attach to a sugar molecule and to a phosphate molecule. And once those attach, once a base pair attaches to the sugar molecule and phosphate molecule, it forms a nucleotide. And these nucleotides form two long strands that spiral into a double helix. That is the shape that DNA takes on. And I did my best to give you all an example of a double helix. It kind of spirals around. If you were to curl a ribbon around my pen, it would form that kind of double helix shape we're talking about. And you should think of a twisted ladder with the base pairs as rungs. So if we were looking at this picture here, this would be our double helix shape that's spiraling around, and each one of these little rungs on the ladder would be a base pair. So we would have adenine and thymine together, and they would be paired up. And then here you would have guanine and cytosine paired up. And then you would have adenine and thymine, guanine and cytosine, all the way down the ladder, where you've got your sugar molecules and phosphate molecules that bound to your base pairs, making up the sides of the ladder here. And you just have this long DNA chain, and then the sequence that you see is going to let you know how that organism builds and maintains itself, because not every creature's DNA is going to be the same. Everyone has a very unique DNA pattern. DNA can replicate or make copies of itself by splitting the ladder in half. So if we're looking at this and we just decided to go through and cut this ladder in half, we would be separating those base pairs all the way down. So then you have AG, AG, and any other base pairs you've got. So we know we've got adenine, guanine, and because we know what's on one side of the ladder, each strand or half of that ladder serves as a pattern for duplicating bases. Since we know we've got adenine here, we know the thing that has to pair with it is thymine. If we've got guanine, cytosine has to pair with it. Since in DNA, adenine and thymine always pair together and guanine and cytosine pair together, you will know, or the cell will know, okay, I've got this DNA, I've only got half of it, but it's a pattern. If I have adenine, I need to add thymine to the other side. If I've got cytosine, I need to add guanine to the other side. So the cell will know, based on that half, that strand that it gets from DNA, how to make a complete DNA molecule and be able to form that double helix and complete the DNA strand. And each new cell needs an exact copy of DNA from the old cell. Because remember, each cell in an organism has the exact same DNA. So if the DNA is replicating itself, it needs to be giving an exact copy to a new cell so that this can continue and the organism is going to continue to have the same DNA in it. So you will see a lot about DNA in biology, and it's important to remember that DNA is the hereditary or genetic material. It's usually found in the nucleus. And remember which bases pair with which. Your straight lines in adenine, A, pair with your straight lines in thymine, T, and your curve G for guanine goes with your curve C for cytosine. DNA is the hereditary or genetic material that is usually found in the nucleus, sometimes in the mitochondria. The most important thing to remember is that it's where all the genetic material is going to be found. It's a big identifier for a cell or for an organism. Enzymes. Enzymes are highly selective catalysts that greatly accelerate the rate of reactions. So they speed up how quickly a reaction takes place. Most enzymes are proteins, but some catalytic RNA molecules have been discovered. So there are RNA molecules that will catalyze reactions and help speed them up in the same way that these proteins will. And so they both work as enzymes. In enzymatic reactions, the molecules at the beginning of the process are known as substrates. During the reaction, these substrates are turned into different molecules known as products. And at the end of the reaction, the enzyme will be unchanged. The enzyme may have to change shape a little bit to bind to the substrate and finish the reaction, but the enzyme itself will be unchanged. It will not have turned into different products. It will not have gotten bigger or smaller. It will remain unchanged. Enzymes work by lowering the activation energy of a reaction, drastically reducing the reaction time. If you had to build up all that energy to complete a reaction normally, it would take a really long time. Well, whenever the enzyme works on a reaction, it lowers the activation energy and makes the reaction happen a lot faster. In fact, a lot of our metabolic processes would not be fast enough to sustain life without enzymes there to speed up the reactions. Enzymes are highly specific for their substrates. That means that each enzyme will only bind with specific substrates that fit with its active site. So each enzyme has an active site. Here we've got our enzyme. To give you an example, this area would be the active site. So both here and here, this open area in the overall shape of the enzyme is the active site, and each enzyme has an active site, and only substrates with a similar shape that would be able to fit in there and bind without the enzyme changing shape too much will be able to fit with that enzyme and complete a reaction. So this would be your substrate. And this is where the substrate would be coming into contact with the enzyme. Here you can see the enzyme and substrate have bonded, and now they have formed the enzyme-substrate complex. And you can see that the enzyme changed shape a little bit to bind to that substrate. And then in this next phase you have the enzyme product complex, where the substrate has started to turn into two different molecules and separate from each other. And then in this last part, you've got your enzyme remaining unchanged, and the products, two separate molecules that the substrate has been turned into, will leave the enzyme. So you can see the enzyme remained unchanged, and the substrate was turned into two separate products. And going through this stage is going to lower the activation energy of a reaction. Enzymes can be affected by inhibitors, which are molecules that decrease enzyme activity, and they can be affected by activators, which are molecules that increase enzyme activity. So where you have enzymes that are raising the rate of speed of a reaction or lowering the activation energy needed, the inhibitors are going to slow that process down, and the activators are going to speed that process up. So where the enzyme is acting on a chemical reaction, these other molecules can also interfere with the chemical reaction and either speed it up or slow it down. The main thing to remember about enzymes is that these are highly selective catalysts, and they are going to increase the rate of a reaction by lowering the activation energy needed, and at the end they are going to remain unchanged. Meiosis. Sexual reproduction in eukaryotes involves a form of cell division known as meiosis. It has several different stages that are carried out to let one diploid germ cell result in 4 haploid gametes. And diploid means that it's going to have multiple copies of the genetic information, where haploid means it's going to have one copy. So, in this first part, this would be interphase. And it's kind of before meiosis starts, but it has to do with it because this diploid germ cell contains genetic information from both a maternal and paternal parents. So you've got female and male genetic material in this one diploid germ cell, and it's getting ready to divide to carry out sexual reproduction. So we have replication going on in here because each chromosome is going to double itself so that there's extra genetic material to carry out this process. In the next phase, we have prophase. And the replicated genetic material is going to line up in homologous pairs, which means that each one is the same genetic material. It's an exact copy. And they're going to cross over a little bit at these points here. And where those cross over, they kind of swap a little bit of genetic material. So each one of these pairs is going to take a little bit of the other pair. And that is called crossing over or recombination. So you're just kind of jumbling up the maternal and paternal bits before the division starts to take place. Then we move on to metaphase. And in metaphase, your centrosomes are starting to pull the homologous pairs apart. And then we move on to anaphase. And you can see they actually are getting pulled further apart. They're not crossing over anymore, and they're starting to move to opposite sides of the cell. And these are all prophase 1, metaphase 1, anaphase 1, because this is kind of a two-step process. Then we keep going, and we've got telophase 1. In telophase 1, there's the cleavage furrow that's preparing for the cells to separate and become two distinct separate cells. There is a nucleus being reformed. The microtubules that have been connecting the chromosomes and pulling them apart have receded, and the genetic material is encased in a nucleus again. And then this last part is cytokinesis 1, where the cytoplasm is split, and there is a definite split made between those two cells. So now there are two diploid daughter cells. So we had our diploid germ cell. The DNA, the genetic material was all replicated, so there was twice as much. We went through this cell division, so now each one of these daughter cells is a diploid daughter cell, and it has twice as much genetic material as it needs to be a haploid gamete. So we're going to go through what is known as meiosis 2, the second step in this process. So we have prophase 2. And in this phase, you'll see we've got the two daughter cells, and now they're kind of repeating the same process that happened up top. So there's no replication because that was an interphase, but they are going to start having the centrosomes move down to either side and start moving out to opposite poles of the cell and start pulling the chromosomes that way. In the next phase, we've got metaphase 2. You can see that the chromosomes have lined up along the equatorial plate in both of these daughter cells. The microtubules are connecting them to the centrosomes, and they're going to start pulling apart. So in our next phase, anaphase 2, you can see that actually happen. The chromosome pairs have gotten pulled apart, and now these individual chromatids are on separate sides of the cell, and they're still going to be kind of being reeled in by the microtubules at this point. And we move on to telophase 2, where we've got a nucleus forming around our new pairs of genetic material that have both the mother and father genetic material in each nucleus. They're all split up now, and we have the cleavage furrow again. The cells are getting ready to divide permanently. Your centrosomes have separated to the opposite sides still, and the microtubules have retracted. And then we go to cytokinesis 2, which is where the cytoplasm divided between these two new cells and these two new cells, and now we've got 4 haploid gametes, which means that they each just have one set of genetic information, and they're going to need a partner to be able to reproduce. So these gametes are going to be ready to be fertilized, and when they are fertilized, a diploid zygote will be formed. So these gametes are ready to be fertilized, and if they are, then they can go on to create a new eukaryotic organism. So meiosis does have a 2-part process, and it goes through the same phases as mitosis, but they're a little bit different because instead of just getting an exact copy, you're mixing genetic material from 2 different organisms, a maternal and paternal parent, and at the end you're going to have the 4 haploid gametes that each has genetic information from both parents. Mitosis. Eukaryotic cells reproduce asexually by dividing into 2 genetically identical daughter cells. Now they do this by the process of mitosis. So let's look. This first phase is called interphase, and it's not really considered a part of mitosis, but it's usually discussed with mitosis because it happens at the beginning right before mitosis happens. It's the build-up to mitosis. It's a period of cell growth and DNA replication. So the cell gets a little bigger, the nucleus gets a little bigger, the DNA replicates, so there's more of it for the mitosis that's about to happen. Chromosomes are in the form of uncondensed chromatin. And the nucleolus may still be present. So this would be our uncondensed chromatin. This darker dot here would be your nucleolus that you could still see. And these little pieces here would be your centrosomes. So whenever you see them along the rest of mitosis, remember these are the centrosomes. Okay, first stage of mitosis is going to be prophase. In prophase, the chromatin condenses into discrete chromosomes. So remember, it had gotten, we had uncondensed chromatin that the DNA had been turned into. In prophase, it condenses into discrete chromosomes. So instead of a bunch of DNA squiggled up, you've got discrete chromosomes that kind of look like little squiggly X's. The nuclear membrane breaks down. So where we had a complete nuclear membrane here, it starts to break down a little bit. And sister chromatids are joined by centromeres. So what they mean by that is this would be your chromosome, and these sister chromosomes or chromatids are joined by these centromeres. So this would be your centromere, and then each of these would be sister chromatids that form the chromosome. The centrosomes are going to move to opposite poles of the cell. So remember, we just kind of had them in the cell here. Now they've moved to opposite sides of the cell, so they're opposite each other and they're kind of equally spaced in the cell. The chromosomes have paired up to form sister chromatids connected by a centromere. And now we're ready to move on to our next phase, which is metaphase. In metaphase, the chromosomes align at the equatorial plate. So where they're kind of just all over the place, they've lined up at the equatorial plate, which means that it's equally distanced from both of those centrosomes that have spaced themselves out at either pole. The microtubules attach sister chromosomes to the centrosomes. So you've got your centrosomes, and these are your microtubules that are attaching the chromosomes to the centrosomes. They've lined them all up along this equatorial plate. And now we're ready to move on to the next stage of mitosis, anaphase. In anaphase, the centromeres divide. So remember, your centromere was that little center here that was kind of holding the two chromatids together. So in anaphase, they divide. The microtubules shorten, so where they're longer, they shorten up, moving paired chromosomes to opposite sides of the cell. So we had these paired chromosomes, and now the microtubules are shortening. They're moving those individual chromosomes over closer to the centrosomes now. And then you're ready for the last phase of mitosis, which is telophase. In telophase, the cytoplasm divides along a cleavage furrow. And so you can see how it starts to kind of dip in. It cleaves in half, and so there's that cleavage furrow, and that's going to show you where your cytoplasm is going to eventually split completely off and form two new cells. The chromosomes condense to chromatin. Remember, that's what we had up here. We had all the squigglies. They're going to just start condensing to chromatin, not a discrete chromosome anymore. And the nucleolus reappears, and the nuclear membrane is going to be reforming around a clear nucleus. So you see the nuclear membrane is forming around where your condensing or your uncondensing chromosomes are, and your nucleolus has reappeared. So once these cells split off, then this is going to be a nucleus again that looks much like this, with the uncondensed chromatin and the nucleolus in the center. Two daughter cells, as we discussed at the beginning, two identical daughter cells will be formed once cytokinesis is complete. And cytokinesis is that process of the cell being cleaved in half to form two new identical daughter cells. So this is mitosis. It is how a eukaryotic cell can reproduce asexually. It just divides into two identical daughter cells. It goes through interphase, prophase, metaphase, anaphase, and telophase. So it starts off with this uncondensed DNA, this uncondensed chromatin genetic material. The chromosomes form little pairs, and the centrosomes come to either side. The centrosomes attach to the centromeres with microtubules, and those chromosomes line up along the equatorial plate. Then they split apart, starting to look like two new cells now, and each part of the chromosome pair goes toward a centrosome so that they are split equally across the cell. And then they split apart, forming a new nucleus with a nucleolus, and cytokinesis is going to be complete once this cell finishes cleaving in half. So just remember that eukaryotic cells can reproduce asexually by mitosis. Nucleic acids. Nucleic acids consist of a linear chain of nucleotides. So that leads us to ask, what is a nucleotide? Well, a nucleotide is going to consist of a nitrogenous base, which could be a purine, such as adenine, which is usually abbreviated with A, and guanine, which is usually abbreviated with G. Another nitrogenous base is a pyrimidine. Another type of nitrogenous base is a pyrimidine, and those would be cytosine, thymine, and uracil. After you get your nitrogenous bases, you also need a pentose, which is a 5-carbon sugar. The most common ones you're going to see are ribose and deoxyribose. And once you have your nitrogenous base in your pentose group, you need a phosphate group. And when you put all three of these together, you get your nucleotides. So let's look at some common nucleic acids. First we have ribonucleic acid, or RNA, which consists of the sugar ribose, so our pentose, our 5-carbon sugar ribose, plus adenine, guanine, cytosine, and uracil. Those would be the nitrogenous bases that you're going to use. And ribonucleic acid, or RNA, is usually single-stranded. The next one would be deoxyribonucleic acid, or DNA, and you notice that Na in both of them is there. It stands for the nucleic acid part. The first letter is telling you which sugar it's going with, which pentose. So deoxyribonucleic acid is going to be with the sugar deoxyribose, plus adenine, guanine, cytosine, and thymine. And you might notice that RNA is going to use uracil as its second pyrimidine, where DNA is going to use thymine as its second pyrimidine. And that's how it always is. RNA is going to use uracil, DNA is going to use thymine. So you need to remember that part. DNA can be single or double-stranded. It is a bit more advanced or complex than the RNA, so a lot of times it is double-stranded, but it can be single-stranded as well, where RNA is usually going to be single-stranded and only rarely double-stranded. Something to remember as well is that adenine always pairs with thymine or uracil, and guanine always pairs with cytosine. And the way I like to remember that is that the curved letters, guanine and cytosine, our abbreviated letters, go together. And then our letter abbreviations that are either made with straight lines, like A and T, go together, or vowels go together. But if you just remember that the curved letters go together, you can always pair up G and C and then pair up whatever's left. And it will be adenine and thymine in DNA, and adenine and uracil in RNA. Genetic information is carried by DNA in all organisms and most viruses. However, some RNA, some viruses use RNA. So, most viruses will use DNA for their genetic material, but some viruses will use RNA. So you can see that nucleic acids are very important for cells. They're made up of a nitrogenous base or series of bases that are going to pair together the same way every time, pentose or sugar, and a phosphate group. And once you get your nucleotide chain and it forms its nucleic acid, you can name it based on which sugar is joined with the rest of it. So ribose formed ribonucleic acid, deoxyribose formed deoxyribonucleic acid, and the nucleic acid's most important function is carrying genetic material. It's going to be carrying the genetic material for all cells and organisms. RNA, or ribonucleic acid, is made up of a base pair connected by sugar molecules and phosphate molecules. Now the bases in RNA are going to be adenine, uracil, guanine, and cytosine. Similar to DNA, they're going to pair with each other, and each one is only going to pair with one other base. They can't pair with any of the other three. So adenine pairs with uracil, and guanine pairs with cytosine. Guanine and cytosine also pair in DNA. Your curved letters for abbreviation pair together, and adenine and uracil pair together in RNA, and it's your vowels that are pairing together. Now in DNA, you have thymine as your other base, the base that pairs with adenine. But in RNA, you've got a different base. So you can think of it as your two abbreviations that are vowels pair together and your two curved abbreviations pair together. So adenine with uracil and guanine with cytosine. Usually, RNA is going to be single-stranded. But it can form a double helix, like the shape of DNA, by folding in on itself. So you have a single strand made up of sugar molecules and phosphate molecules, and then you've got the little base pairs coming off of it. So it may be like this. You've got your sugar and phosphate molecules, and then you've got your little base pairs coming off of it. And my little lines here are splitting it. So you would have adenine and uracil, guanine and cytosine, adenine and uracil, guanine and cytosine, all the way down. Now, if the RNA was more complex, it may fold in on itself, and this could curve around and kind of form a double helix by being tightly coiled and wrapped together, so that instead of having just one side of the ladder, it curved up and made the one side kind of sandwich it together and form into a double helix that way. But it's not going to be the same kind of pattern, the same structure as DNA is going to be. RNA is also a carrier of genetic material in some viruses. So in viruses, RNA can carry the genetic material, and a lot of times if it's a more complex virus, you'll have RNA that is a double helix. It has formed a more complex structure to carry genetic material for the viruses. In most cells, DNA is what's holding all of the genetic material, but RNA is what kind of moves it around the cell. So let's look at the three main types of RNA. Messenger RNA, or mRNA, carries codes from DNA in the nucleus to ribosomes in the cytoplasm. So it's moving, it's being a messenger and carrying that code from the DNA to ribosomes. Ribosomal RNA, or rRNA, they read that code carried by the messenger RNA. And ribosomes are composed of the ribosomal RNA and proteins. So the ribosomal RNA is a big part of ribosomes. And then transfer RNA, or tRNA, bring amino acids to the ribosomes where they are linked to proteins. So you can see RNA is moving around the cell a lot. It's not just staying in one place. DNA is mainly just going to stay in the nucleus and hold that genetic material. RNA is going to move it around, except in the case of viruses, where RNA is the main holder of genetic material instead of just a transport system. So let me show you what this might look like. So let's say we've got our ribosome, and so you've got RNA, your ribosomal RNA in here. Well, you've got DNA in the nucleus over here, and it's going to get transferred to RNA via the messenger RNA. So it's going to send that code along, and it's going to come into the ribosomal RNA. And then you've got transfer RNA out here, and it's going to bring amino acids into the ribosome. So you've got your ribosome here, and your ribosome holds the RNA. It takes in the mRNA, reads the code from DNA, it takes in the transfer RNA and picks up these amino acids, and out comes a protein. So the whole point of all this was to create a new protein, and RNA is a big part of protein synthesis. So it's going to move the code from the DNA via messenger RNA into a ribosome, and transfer RNA is going to bring amino acids into the ribosome, and the ribosomal RNA is going to take the code and take the amino acid and create a protein and spit it out. So that's what's going on here. Remember the big differences between RNA and DNA? DNA is holding all that information in the nucleus. RNA is moving the information around. It's how DNA gets its code from one place to another. So RNA is more of a transfer system within the cell, and it has several different kinds of RNA so that it can do different jobs in the cell, all working on transferring that genetic code.
We're going to take a look at some parts of cell anatomy, starting with the nucleus, which contains the chromosomes and regulates the DNA of a cell. Now, the nucleus is responsible for passing on genetic traits between generations. The nucleus is a small structure and really is the defining structure of eukaryotic cells. And all eukaryotic cells have a nucleus. Now, chromosomes are highly condensed, thread-like rods of DNA. And DNA is the genetic material that stores information about the plant or animal. So since chromosomes contain DNA, then really chromosomes can contain or store the information about the plant or animal. Now we move on to chromatin, which consists of the DNA and the protein that makes up chromosomes. So there you can see a direct relation between chromosomes and chromatin. Now when we look at the nucleolus, this is involved in protein synthesis and synthesizes and stores RNA, which is ribonucleic acid. So the nucleolus is the structure contained within the nucleus, and so it consists of protein. It is small, round, doesn't have a membrane, but it's involved in what we know as protein synthesis and synthesizes and stores RNA. And so you notice here that the nucleolus, nuclear pores, nuclear envelope, and nucleoplasm all look very similar to the word nucleus. So these last four parts of cell anatomy that we're covering in this session all directly relate to the nucleus, because the nucleolus is contained within the nucleus. And then we have nuclear pores, which regulate the exchange of material that goes on in a cell. It regulates the exchange going on between the nucleus and the cytoplasm. Then we have the nuclear envelope, which consists of inner and outer membranes made of lipids, and it is basically a structure that encloses the structures of the nucleus. And then we have nucleoplasm, which if you're familiar with cytoplasm, it's very similar to that. in the way it sounds, and that's because it's the liquid within the nucleus, and so it's very similar to cytoplasm. So that's a look at some of the parts of cell anatomy, especially those parts that relate directly to the nucleus. We're going to take a look at some parts of cell anatomy, starting with cytosol, which is liquid material in the cell. It is mostly water, but it also contains some floating molecules. And then we have cytoplasm, which is a term used to cover the cytosol and the substructures found within the plasma membrane. So the cytoplasm is a collective term for the cytosol, which is the liquid material in the cell, and the substructures, or we could say the organelles found within the plasma membrane. And now we come to the plasma membrane, which can also be called the cell membrane, which defines the cell by acting as a barrier. And so the cell membrane helps keep cytoplasm in and substances located outside the cell out. It also determines what is allowed to enter and exit the cell. And then we come to endoplasmic reticulum, which is the tubular network that comprises the transport system of a cell. Now there's two types of endoplasmic reticulum. The first is rough, meaning it has ribosomes on the surface, and smooth, meaning it does not have ribosomes on the surface. It is fused to the nuclear membrane and extends through the cytoplasm to the cell membrane. And then we have mitochondrion, which generates ATP and is involved in cell growth and death. Now, the cell structures vary in terms of size and quantities. So some cells may have one mitochondrion, while others have thousands. And so mitochondrion performs various functions like generating ATP and being involved in cell growth and death. So that's a look at some parts of cell anatomy. We're going to take a look at some parts of cell anatomy, starting with ribosomes, which are involved in synthesizing proteins from amino acids. Now, ribosomes are numerous, making up about 1/4 of the cell. Some cells even contain thousands of ribosomes. Some of these ribosomes are mobile, and some are embedded in the rough endoplasmic reticulum. And then we have the Golgi complex, which can also be called the Golgi apparatus. And this is involved in synthesizing materials such as proteins that are transported out of the cell. Now, the Golgi complex is located near the nucleus and consists of layers of membranes. And then we have vacuoles, which are basically sacs used for things like storage, digestion, and waste removal. And then we have vesicles, which are small organelles within a cell. They have a membrane and perform varying functions, such as moving materials within a cell. And then finally we have the cytoskeleton, which consists of microtubules that help shape and support the cell. So that's a look at some parts of cell anatomy. We're going to take a look at some parts of cell anatomy, starting with microtubules, which are made of protein and are part of the cytoskeleton. And they also help support the cell. And then we have the centriole, which is involved in cellular division. Now, the centrioles are cylinder-shaped structures near the nucleus. Each cylinder consists of 9 groups of 3 microtubules, and the centrioles occur in pairs. And then we have the centrosome, which has a unique relationship to the centriole because it is comprised of the pair of centrioles located at right angles to each other and surrounded by protein. And so the centrosome is involved in mitosis and is also involved in the cell cycle. And then we have the lysosomes, which digest proteins, lipids, and carbohydrates, and it also transports undigested substances to the cell membrane so they can be removed. Now, the shape of a lysosome depends on the material being transported. So it digests some things, and things it can't digest, it transports. And then we have cilia, and the plural form of cilia is cilium. And so the cilium is appendages extending from the surface of the cell. And so these appendages cause the cell to move. And the movement of these appendages can also result in fluid being moved by the cell. So that's a look at some parts of cell anatomy. We're going to take a look at some parts of cell anatomy, starting with flagella. And flagella use whip-like movements to help the cell move. These are tail-like structures on cells, and so the flagella use a whip-like movement in order to propel the cell. Now, these are similar to cilia, but are usually longer and not as numerous. A cell is generally only going to have one or a few flagella. Then we have the cell wall, which provides plants with a sturdy barrier. And this barrier can hold fluid within the cell. This cell wall is going to be made of cellulose and composed of numerous layers and surrounds the cell membrane. Then we have chloroplasts, which plant cells use for photosynthesis. Now, if you're not familiar with photosynthesis, this is the process plants use to create food energy from sunlight. Now, chloroplasts are specialized organelles, and chloroplasts also contain chlorophyll, which has a green color. Then we have plastids, which are used to make chemical compounds, and they're also used to store food. And so plastids are membrane-bound organelles found in plant cells. And plastids can also contain pigments used during photosynthesis. So plastids, like many of these other parts of a cell, can serve kind of double duty. They have a couple of functions that they contribute to. And then we have the plasmodesmata, which has a really long name, but this is the name for the channels between the cell walls of plant cells, and they allow for transport between cells. So that's a look at some parts of cell anatomy.
Today we're going to go over combustion and terms and concepts related to it. So if you look at the board behind me, we'll just start at the top here. When we talk about autoignition temperature, when we think about autoignition temperature, we're talking about the temperature at which a substance is so hot that it just bursts into flame without need for an ignition source. As things get hotter and hotter, most substances, though not necessarily all, have a point at which when they reach that temperature they will, if there is oxygen or other medium in which it can burn, burst into flame on its own without the need for an external source of combustion, heat, fire, anything like that. It just on its own, spontaneously. And so it would be tied into number 5 here, spontaneous combustion, no source of ignition. When we think about things that don't need a source of ignition and yet burst into flame, we can think in terms of maybe a compost pile. As the compost pile sits there, it's got hay and other things in it probably. It's strongly insulated, and there's fermentation going on inside of it through the bacteria, which is generating heat. And as the heat rises because of the insulation, there's nowhere for it to go. It eventually reaches a temperature at which ignition can take place without an outside source, and it bursts into flames. And so sometimes people do have compost piles that go up in flames because of the spontaneous combustion medium, the fermentation by the bacteria, raising the temperature, it's well insulated, and then boom, up it goes. When we think about combustion, we have certain types of reactions. Exothermic reactions are those that give off heat. They generate heat and force it out, sometimes light, sometimes noise in the form of explosions, but exothermics give off. Endothermic absorb or take in heat in these kind of reactions. So there is a heat transfer taking place here. Exothermic forcing it out, endothermic pulling it in, absorbing heat inward. When we think of endothermic reactions here, you can think of these sports medicine instant cold packs that has a chemical reaction inside them that gets very cold very quickly. This is an endothermic reaction. So when we think about heat transfer, which is very much a part of combustion, we need to remember that what's going on is within the way physics works now, thermal equilibrium is always trying to be sought. So if there's something that's warmer next to something that's colder, the general motion will be that the warmer temperature will move towards the colder temperature until these reach equilibrium. If you think here of a rod that's been superheated on one end and is room temperature on the other end, what's going to happen then in conduction with this thermal transfer is the kinetic energy from the rapidly moving molecules and atoms down here on the hot end running into the colder ones, transferring their heat and energy that way. And so the heat moves down from the hot end towards the cold end, eventually reaching a place of equilibrium where this loses its heat, this gains the heat, and then they match. So heat transfer is very much a part of combustion. Conduction being one of those things, direct molecular transfer of heat until thermal equilibrium is reached. Another form of heat transfer is radiation. This is electromagnetic wave transfer of heat. Here we need to think in terms of the sun, which gives off electromagnetic waves or radiates radiation, and it heats the earth. It communicates it through the medium of a vacuum, space, but it still arrives here in the form of heat transfer, radiation, as it warms the earth and strikes the earth. So that would be a form of heat transfer radiation. And then convection. Convection is heat transfer through a fluid medium. Now when we say fluid, we don't automatically mean liquids. Fluids are also gases. So a fluid medium is a gas or a liquid. So you can get convection currents in air as well as in water. What you have then is heat is applied to the fluid, either gas or air, and as the fluid is heated, it gets less dense and warmer, and so it rises, causing the more dense, colder fluid then to move downward, and you get a circular motion. If we think in terms of boiling water, you can actually watch as it seems to churn up from the bottom. The heat source is there. The hot water becomes less dense, lighter, comes to the surface. The cold water sinks down, is heated, and comes up again. You can think in terms of air as well. Hot air, less dense, lighter, rises, cold air, more dense, heavier, sinks, is heated, and sent around. So you get these circular convection currents. So these are the main things to keep in mind around heat transfer: conduction, direct molecular radiation, waves, convection, this circular motion through a fluid medium, gas or liquid. We talked about spontaneous combustion in your compost pile under auto ignition temperature, and then we've got hypergolic reactions here, or combustion. Basically that's a substance when brought into contact with another substance that immediately ignites without an ignition source necessary. It's just that type of reaction, and this is what they do in rocket engines. They have two separate substances kept in different compartments, and then they are injected into a third compartment at the same time, and when they meet and react, you get combustion automatically without an ignition source, which then they use to propel the rocket upwards. So hypergolic substances are those that ignite or combust when brought into connection with an oxidizer. So this substance brought into an oxidizer immediately combusts, and of course you get rocket explosions directed beneath the rocket, which sends it up. So hypergolic combustion. Well, this has just been a brief overview then of things related to combustion, some of the concepts, some of the vocabulary in those concepts. If you'd like to learn more about this and related matters, if you'll look underneath this video, there's a link down there. And if you'll click on that link, it'll take you to the website that has that information. And while you're on that website, you'll also find a link to an ebook that's ready for immediate download.
Mendel's laws consist of two laws pertaining to genes. The first being the law of segregation, which says there are two alleles and half of the total number of alleles are contributed by each parent organism. So that makes sense. There's two parents and each parent does half of what needs to be done as a whole. And so they each care for their half of the load. And the second law is the law of independent assortment, which says traits are passed on randomly and are not influenced by other traits. Now the exception here is linked traits because these are not passed on randomly. Now a Punnett square goes along with Mendel's law because a Punnett square illustrates how alleles combine from the contributing genes to form various phenotypes, which a phenotype is the collection of all visual genes. So the Punnett square combines each set of parent genes and puts them in columns and rows, and that way you can see all the possible allele combinations. And so that allows a person to get an idea of what type of phenotype the offspring organism is going to have given the genes of the parent organisms. So a Punnett square is also important to understand. So again, Mendel's laws are the law of segregation and the law of independent assortment.
The brain is one of the most important parts of our nervous system, and today we're going to go over the various parts of the brain and talk about some of the ways they contribute and help in the areas over which that part of our brain control. And so we're going to begin then with the frontal lobe. The frontal lobe controls our emotions, our judgments, and our motor aspects of our speech, and is also the primary motor cortex for voluntary muscle activation. After the frontal lobe, we have the parietal lobe, which receives sensory information from touch, proprioception, which has to do with our ability to tell where our arms and legs are and our orientation in space. So even with your eyes closed, you know where your arms and legs are. You have a sense of your body. This is proprioception. The parietal lobe helps govern that. Also our sensation of temperature and pain. That's the parietal lobe. Then the temporal lobe is responsible for our hearing, auditory information, and our language comprehension. The occipital lobe is our center for visual information, our sight. The cerebellum coordinates all of our muscle function. And then we have the brain stem, and within the brain stem we've got the midbrain, the pons, and the medulla. The brain stem governs our breathing, our respiration, our hearts, our cardiac center, and all the nerve pathways to the brain come through the brain stem. And then finally, moving on then from that to the diencephalon, which is the thalamus, subthalamus, and hypothalamus. The thalamus helps integrate and relay sensory information from the face, feelings there, our retinas, our cochlea from our ears, our taste receptors, and interprets sensation of touch, pain, and temperature. So our thalamus is very important to not only our sense of what we feel in terms of pain, temperature, touch, but also our retinas, our eyes, our cochlea, and our ears, and our taste and our tongue. The hypothalamus controls the autonomic nervous system and the neuroendocrine system. It maintains our body homeostasis, helps regulate body temperature, helps regulate our appetites, governs our thirst and how we feel that, our sleeping cycle, and hormone secretions. So once again, the brain, very integral, necessary, core part of our nervous system, broken up into these various regions and what they controlled. If you'd like to find out more about the brain and its relation to our nervous system, I encourage you to go to the link that you'll find below this video. At that website, you'll find an ebook that is ready for immediate download.
Water is essential to life as we know it. and it is one of the main, or the main constituent of many living things. And so there's several interesting properties of water I want to take a look at. So first is its high polarity. It has negative and positive ends. And this is due to the fact that water is made up of 2 hydrogen atoms and only 1 oxygen atom. Then also hydrogen bonding. Water molecules engage in hydrogen bonds with other water molecules. Water also is cohesive. And so cohesiveness means it's attracted to itself. You may have noticed this if you ever filled a cup of water up a little bit too high above the rim of the glass. Well, the water didn't immediately spill over because of cohesiveness. The water molecules were attracted to the other water molecules, so the water didn't necessarily spill out because the attraction was stronger than the force of gravity trying to pull it out. Then we also have adhesiveness, which means water molecules are attracted to other molecules. Water has a high specific heat, which means it resists the breaking of its hydrogen bonds and resists heat and motion. So specific heat means that it has a large amount of heat required to raise temperature by a specific amount. This also kind of goes along with high latent heat, which is the net heat change in change of state. So basically when water changes from one state to another, that means there was a lot of heat either absorbed or put off. And so net heat change means how much water was absorbed or exerted to do this change of state. Now, water in its solid form as ice floats in its liquid state of water. Now this is pretty odd because many times substances are denser in their solid forms, but water is strange in that its solid state floats in its liquid state. This is because when water is frozen, it expands, and so it becomes less dense because it's taking up a larger amount of space. So that same material is spread out over a larger amount of space, which means it's less dense than the liquid water. And so when anything is less dense than water, then it's going to float in that water. So that's why ice cubes float. So that's a look at some of the interesting properties of water. Scientists throughout the years have had different ideas of what the atomic model looked like, or what the structure of an atom was. So scientists throughout the years came up with these different ideas, often building on past scientific discoveries by modifying those discoveries. So I want to take a look at the discoveries of 5 scientists in terms of what they thought the atomic model looked like. So we start with John Dalton, who thought of atoms as small, solid, individual balls. So he thought of them as small, solid, individual balls. And then there was J.J. Thomson, who introduced the plum pudding idea. Now, today, we don't accept the plum pudding atomic model as accurate, but it did get the atomic model on the right track to being accurate. Upon discovering the electron, J.J. Thomson came up with this plum pudding model. Like raisins in an amorphous pudding, electrons were embedded in a positive sphere, or so he thought. Then we come to Ernest Rutherford, who thought of, or came up with, the planetary model. He thought of atoms as being mostly empty space, and that he believed that most of the mass of an atom came from the small nucleus. Electrons were found orbiting the nucleus, so that's why he called it the planetary model, because electrons were orbiting the nucleus like planets orbit the sun. Then Bohr came up with the idea, building on the planetary model or modifying the planetary model, to say that the electrons orbited the nucleus in discrete circular energy levels or shells. So you can see here how Bohr didn't come up with this idea on his own. He took the planetary model and modified it some to be up to date with some current scientific discoveries. So notice this was discovered by Bohr, so we call this the Bohr model, or the Bohr atomic model, which you are probably somewhat familiar with. Then finally, we come to Erwin Schrödinger, who came up with the idea that electrons orbited in clouds. So this is a little bit different than the other ideas. See, Schrödinger showed that the electrons orbited the nucleus not in discrete orbits, but rather in clouds, and the electron clouds had various shapes and described the probabilities of the electron locations. The electron cloud model is the current quantum model of the atom. So up here on the board, you can basically just see how atomic models evolved with the more discovery that scientists had to come to the model that Schrödinger came up with, which is the current quantum model of the atom. In other words, it's the most accurate model that we have. A covalent bond is formed between two or more atoms that share pairs of valence electrons. Now, a single covalent bond is just one pair of shared electrons. Water, which is H2O, has two hydrogens to every one oxygen. Each hydrogen atom requires one valence electron to fill its outer shell, and oxygen requires two valence electrons to fill its outer shell. So, by sharing one valence electron with each hydrogen atom, oxygen fills its outer shell. Likewise, by sharing their valence electrons with oxygen, each hydrogen atom fills its outer shell. So, in water, oxygen has two covalent bonds, one to each hydrogen atom. So this looks something like this. You have oxygen with its inner shell, and then it also has an outer shell. So you have two electrons in its inner shell, and then oxygen has 6 total electrons in its outer shell. But remember, it needs 2 more valence electrons to fill its outer shell. And hydrogen looks something like this with 1 electron, and there's 2 hydrogen atoms just like that with 1 electron that need 1 more valence electron to fill their outer shell. So what happens here in covalent bonds is hydrogen comes over here and donates its valence electron to oxygen. Oxygen donates one of its valence electrons to hydrogen. So basically they're sharing these valence electrons. And same thing happens with this other hydrogen atom. And due to my drawing inabilities, this doesn't look very good, but these still are circles over here. And what's happening is hydrogen and oxygen are sharing these valence electrons so that both of their outer shells are filled. And then you have 2 covalent bonds here. You have one here and one here because a covalent bond is 1 pair of shared electrons. And there's a pair of shared electrons here and a pair of shared electrons there. Now I want to go over some things that are generally true about covalent bonds, but I'm not going to present these as facts because they're not facts and they're not laws. They're not always true. They're just generally true. And so covalent bonds generally form between nonmetals. And if you look to our example, here we have 2 covalent bonds formed between nonmetals because oxygen and hydrogen are both nonmetals. And then covalent bonds generally have lower bond energies than ionic bonds, which a bond energy is the amount of energy it takes to break a bond apart, which the energy is usually in the form of heat. And then the atoms of similar electronegativity value usually form covalent bonds. So the atoms that make covalent bonds up have similar electronegativity values. So those are some general facts about covalent bonds. If you just remember a couple of things from this session, remember that covalent bonds are formed between 2 or more atoms that share pairs of valence electrons. And a single covalent bond is just 1 pair of shared electrons. Ionic bonds tend to form under two certain criteria. And the first is ionic bonds tend to form between metals and nonmetals. So if you have two elements and one's a metal and one's a nonmetal, an ionic bond is likely to form. Now the second criteria is that ionic bonds tend to form between elements with a large difference in electronegativity. So the larger the difference in electronegativity, the better the chance ionic bonds are going to form. And so if elements meet both of these criteria, there's a very good chance they are very likely to form ionic bonds. Now, keep in mind that elements with the highest electronegativity values are in the upper right-hand corner of the periodic table of elements, while those with the lowest are near the lower left-hand corner. So you have elements with low electronegativity down here, and elements with high electronegativity up here. And so just by knowing those two facts right there, if you see an element on the periodic table, if you just imagine this board as the periodic table, and it's about right here, you just know by where it's geographically situated on the board that there's a really good chance that it has a high electronegativity because it's near the top right-hand corner of the periodic table. Now, I have some pairs of elements here, and I want to examine these elements to see if they meet this criteria and determine whether or not they are likely to form ionic bonds. So first we have nitrogen and oxygen. Now, nitrogen is a nonmetal, and then oxygen is obviously a nonmetal. So they are not likely to form ionic bonds because there is not between a metal and a nonmetal. We have two nonmetals here. Then we have potassium and fluorine, and potassium is an alkali metal, and fluorine is a nonmetal. So we've met the first criteria. We have reaction here between a metal and a nonmetal. So we're doing good so far. And then there's a high electronegativity difference here, and it's actually 3.2, which is pretty high. So they're likely to form ionic bonds, specifically potassium chloride. Now here we have barium and sulfur, and barium is a metal, and then sulfur is a nonmetal. So here we have a metal, a nonmetal. We're doing good so far, so they're likely to form ionic bonds, specifically barium sulfide. And then in addition to that, their electronegativity difference is moderately large at about 1.6. Now finally, we have cesium and tin. And cesium is an alkali metal. And then I'm sure you can guess that tin is a metal. So here we have two metals interacting with each other, so it's not between a metal and a nonmetal. So these cesium and tin is not likely to form ionic bonds. So notice here the main factor here was that we had two nonmetals and two metals here. But when we have a metal and a nonmetal, they're likely to form ionic bonds, and especially if they have a high electronegativity difference. So hopefully through this short session, you have a better understanding of ionic bonds. Some common types of lipids that you are probably familiar with are fats, oils, and waxes. Now, like carbohydrates, lipids are organic molecules. However, lipids have a more complex structure than carbohydrates and are larger molecules. Now, lipids include several different types of groups. And so the first is triglycerides. And so stated somewhat simply, triglycerides consist of a glycerol molecule that is bound to 3 long-chain fatty acids. So glycerol is a polyalcohol, and fatty acids are long-chain carboxylic acids. So triglycerides are the most common lipids and make up fats and oils. Now, a second grouping is phospholipids. And these consist of two fatty acids bound to a phosphate head group. And so the fatty acid portions avoid water, meaning they're hydrophobic, while the phosphate head groups seek water, meaning they're hydrophilic. So phospholipids have a structural function. They make up a cell and organelle membranes in your body. A third group is steroids, which are lipids that are derived from cholesterol. Steroids have a four-ring structure with a hydroxyl group at one end and a short carbon chain at the opposite end. Cholesterol is also found in cell membranes and circulates in the blood. Another type of steroids are hormones. Now, the fourth group of lipids that we're going to talk about are waxes, which are formed when a long-chain alcohol binds to a long-chain fatty acid. So again, some common types of lipids are fats, oils, and waxes. And like cholesterol, lipids are organic molecules. The periodic table is a way to systematically display the chemical elements. Now notice I said that it consists of chemical elements, and it consists of elements only. So this does not include mixtures and compounds, because an element is basically matter in its most basic form. The periodic table positions chemical elements based upon atomic number. An atomic number is the amount of protons an atom has in its nucleus. So hydrogen is the first element on the periodic table because it has one proton in its nucleus. And then the next chemical element has more protons in its nucleus. Like I said, the periodic table arranges chemical elements very systematically. So if you were to take a look at one row on the periodic table, all the chemical elements on that one row would have the same number of electron shells. And if you were to take a look at one column, all of the chemical elements in that column would have the same electron configuration. So the periodic table arranges the elements in such a way that all the elements are next to similar elements. So if a scientist knew a lot about one element, he could know a lot about the elements around it just because all the elements are grouped together with similar elements. There are about 118 elements out there, but only 114 have been officially recognized. And 98 of those are natural elements, ones that naturally occur in nature, such as potassium, nickel, and hydrogen. The other ones are synthetically produced by humans, such as Einsteinium. So the important thing to remember about the periodic table is that it consists of chemical elements, and it is arranged by atomic number. pH is basically a measure of how many hydrogen ions are in a substance. pH is very important to many fields such as biology, chemistry, medicine, food science, farming, and water treatment. I've written up here on the board a pH scale ranging from numbers 0 to 14, each indicating the amount of pH in a substance. Now there are pHs that go higher than 14, but for practical purposes, this is the largest pH scale we need. Water has a pH of 7. And any substance with a pH larger than 7 is considered a base. So everything from 7 over to 14 is considered a base. Common bases that you probably know of are baking soda and bleach. Now any substance with a pH below 7 is considered an acid. Some of those that you may know of are lemon juice, tomato juice, and gastric acid. So water is kind of the middle bar on the pH scale, and anything to the right is the base, and anything to the left is an acid. The three classical states of matter are solids, liquids, and gases. So first we're going to look at solids, and we're going to look at some of the characteristics of a solid. So solids have a definite volume and density. So we would say they have a definite volume and density at a given temperature and pressure. So like this whiteboard, for example, is a solid. So the volume of this whiteboard isn't changing, and the density of this whiteboard isn't changing. And then the second point of a solid is it has a degree of structural rigidity and a constant shape. So notice that this whiteboard has structural rigidity and a constant shape, so the shape isn't changing. It also has a resistance to flow. Now, some solids, such as modeling clay, can flow and undergo deformation under pressure. So I'm just going to write here, there are exceptions, but for the most part, solids have a resistance to flow. Now, the second state of matter is the liquid, and like solids, at a particular temperature and pressure, it has a definite volume and density. So the reason I say at a certain temperature and pressure is because at a different temperature and a different pressure, this whiteboard may have a little bit of a different volume and a little bit of a different density. But as long as it stays at the same temperature and pressure, then it's going to stay the same. The volume and density are going to stay the same. The same thing with the liquid. Now, a liquid does flow readily, so that makes it a lot different than a solid. But it does not expand to fill a container. So it's going to flow readily. If you spilled water on the ground, it wouldn't all just stack up in one spot. It would flow all over the place. Well, if you were to pour water into a glass, the water isn't going to expand to fill up that whole glass. It's going to keep the same volume. Now, that's different than the third state of matter, which is a gas, because the main thing about a gas is that it will expand to fill a container. So all of a sudden, if a gas was exposed in this room, it wouldn't all stay in one spot. It would eventually move to fill the entire room. Now, the molecules are spread much farther apart, and they move more rapidly and randomly than in a liquid. So basically, the molecules move the slowest in a solid, they move a little faster in a liquid, and then they move the fastest in a gas. And so that's why gases are spread apart. It's because they're moving around so much and bumping into each other and pushing away from each other. And a liquid has more movability to it, I guess, if that's a word. It can move more because the molecules move more. And then a solid, you think about ice, it's not really doing anything. It's just sitting there because it's a solid. The molecules are moving really slowly. And so a gas is far more dispersed than even a liquid because it'll expand to fill an entire container. And so that's a look at the three states of matter. We're going to take a look at strong acids and weak acids, as well as strong bases and weak bases. Now, strong acids and strong bases obviously have their differences because one's an acid and one's a base. But they do have two things in common, and that's that strong acids dissociate completely in water, 100%. And they are also strong electrolytes. Now, an example of a strong acid is hydrochloric acid, and an example of a strong base would be sodium hydroxide. Now, like strong acids and strong bases, weak acids and weak bases have their obvious differences, but they do have one thing in common, and that's that they do not dissociate completely in water. So just stick with me here so you can remember the differences, that strong acids and strong bases do dissociate completely in water, but weak acids and weak bases do not dissociate completely in water. An example of a weak acid is hydrogen sulfide, and an example of a weak base is ammonia. So that's a quick overview of strong and weak acids and strong and weak bases. The scientific method is a process that has been around for over 1,000 years that is used by scientists to make scientific discoveries. There's a progression in the scientific method. The scientist starts by asking a question that the scientist wants to have answered. In this case, the question is going to be, which cat is faster? The scientist is interested in knowing if the lion or the cheetah is faster. Then the scientist, based off of prior knowledge, is going to formulate a hypothesis. In this case, the hypothesis is going to be the cheetah is faster. Notice how the hypothesis is written as an established fact. It's because through the experiment portion of the scientific method, the scientist is going to try to prove his hypothesis true. Then comes the prediction portion of the scientific method. In this portion, the scientist is going to try to clarify his hypothesis. So in this case, the prediction is going to be the cheetah will be faster over a 60-meter period. It's important to realize the difference between a hypothesis and a prediction. A hypothesis is stated as established fact, and the experiment is either going to prove or disprove the hypothesis. The prediction clarifies the hypothesis because otherwise the experiment could just prove that the cheetah is faster over a 2-meter period, but that doesn't really show any endurance on the cheetah's part. So the prediction says that the experiment results are going to show that the cheetah will be faster than the lion over a 60-meter period. Then comes the test portion of the scientific method. This is very important. This is the experiment part of the scientific method. This is when the scientist either proves or disproves his hypothesis. So I'm going to write here that the scientist is going to use a radar gun. So the scientist is going to point the radar gun at a cheetah running over a 60-meter period and write down how fast the cheetah was running, and then point the radar gun at a lion running over a 60-meter period and write down the speed of the lion. Then comes the analysis portion of the scientific method. This is when the scientist looks at the results and decides whether they prove or disprove the hypothesis. So in this case, the analysis is going to prove and confirm the hypothesis. So I'm going to write here that the hypothesis is confirmed, because the results of the experiment showed that indeed the cheetah was faster than the lion. So if my example confused you a little bit, don't worry about it. The important thing to remember is the main points of the scientific method: the question, hypothesis, prediction, test, and analysis. Specific heat capacity can be shortened to just be called specific heat, and we can abbreviate that as a capital C. And that's the amount of heat energy that it takes to raise the temperature of 1 gram of a substance by 1 degree Celsius. So it's a measure of a substance's ability to store and transfer heat energy. Each substance has a unique specific heat. Water has a relatively high specific heat, meaning that it can absorb or release large amounts of heat with a little change in temperature. In contrast, copper has a low specific heat. It absorbs or releases a small amount of heat with a large change in temperature. Now, you measure a substance's specific heat in a calorimeter. And this is where heat is transferred from a known mass of the substance to a known mass of water. The corresponding temperature changes of the water and the substance are then measured. So the equation for specific heat is this right here. Specific heat equals heat transferred divided by mass times change in temperature. So let's go ahead and dive in and take a look at an example problem. A 100-gram copper block absorbs 1,170 joules of heat to raise its temperature by 30 degrees Celsius. So the question is, what is the specific heat of copper? Now, since we're looking for the specific heat here, this is a direct application of this equation right here. And so all we have to do is fill in the appropriate numbers. And so first we're looking for the change in heat. So that would be 1,170 joules divided by the mass, 100 grams. And then times the change in temperature, which is 30 degrees Celsius. So I'll save you the math on this. The answer is 0.39 joules divided by grams Celsius. Now, let's go ahead and take a look at a more difficult example problem. How much heat is required to change the temperature of a 25-gram block of iron by 100 degrees Celsius? And then we're given the specific heat of iron. And so here we're looking for the change in heat. So we're looking for Q, so we have to rearrange the equation to solve for Q. So here, to solve for Q, we're multiplying the mass times the specific heat of iron times the change in temperature. So now we just need to plug in the numbers. So we're dealing with 25 grams times the specific heat, which is right there, so 0.45. Then finally, we multiply it by the change in temperature, which is 100 degrees Celsius. And so then again, I'll save you the math, but the answer is 1,125 joules. And so the units here is just joules because we're looking at heat, and that's because Celsius and grams can both be crossed out there. So we're left with joules, which is the unit we need. So again, specific heat is the amount of heat energy that it takes to raise the temperature of 1 gram of a substance by 1 degree Celsius.